So this is a slice of an actual human brain, which is the most complicated and the most versatile computer that we know. It isn't the best computer these days for crunching really big numbers or for playing chess or even for playing Jeopardy, but it's terribly versatile. It's capable of unlimited creative thought, of artistic appreciation, of artistic creativity. It's capable of moral judgments, and it's capable of personal empathy. My objective tonight is to tell you something about how this apparatus works, the general rules for the way in which the nervous system sends and receives its signals. And then, since many of you are parents, I want to focus in particular on how it gets put together. What are the processes that are involved in elaborating this remarkably complex organ and seeing that it's put together properly? So, to begin, I just want to remind you of the structure of the brain. As you know, we have a spinal cord which ends in a brainstem, and this is the region that is responsible for automatic functions such as breathing, control of the heart, control of digestion, and whatnot. Above that lies the cerebellum, which is involved in coordinated motions, athletic endeavors, anything that requires fine balance or fast activity. And everything else on the top is the cerebrum, which has a frontal lobe, parietal lobe, occipital lobe, and temporal lobe, of which I'm sure you've heard. This is where we live. This is where the complex human activities reside. This is where we sense our environment. This is where we make decisions. This is where we take actions. And most of the presentation will be about that. Now, there's a daunting problem that one confronts in thinking about the nervous system, which is that there's something on the order of 100 billion nerve cells in our brains. There's something on the order of 100 trillion connections among all of those. So it's such a bewildering amount of complexity that it seems almost impossible that we understand how it works. But as I'll show you in a few minutes, the basic underlying principles are actually rather few in number and are quite accessible. The other point that I'll be dealing with is the fact that all of this apparatus has to be specified by only about 20,000 genes. We're still counting genes. It might be 30,000. Some people think it might be 40,000. But this is a paltry number by comparison to the other two. How can it be that this very small amount of genetic information can accurately and repetitively specify an organ as complex as the human brain? This gives you a sense of the complexity I'm talking about. This represents about 1/billionth of the brain's complexity. That is about 100 nerve cells. And you can see that these nerve cells have very complicated individual personalities, all kinds of branching, all kinds of elaborate interactions that are a bit bewildering. And even isolated individual nerve cells have this complex series of processes hanging off of them, the Christmas tree sort of arrangement, or this large, flat espalier sort of arrangement. Again, how is all of this put together? So the first general principle is that all the activity stems from that of nerve cells, from neurons. And neurons have only a few very specific parts. Like every cell, a neuron has a cell body. And in that cell body is a nucleus which has DNA in it. That's where the genetic information, all of the wiring diagram for the cell is stored. Neurons are special because they have these antennae called dendrites that are processes emanating from the cell body and radiating out in all directions. And as you'll see in a moment, this is the site at which incoming information is received by the nerve cell. They also have an axon, which is a process that can conduct a particular type of electrical signal down to its end. And again, I'll show you what that signal is in just a moment. And finally, they have a synaptic terminal where that information is transmitted to the next set of cells in the network. So there's a flow of information into this end of the cell, along the axon, and finally transmission to cells that lie beyond. The signals that we are dealing with have to do with ions and membranes. And as you will remember, every cell has got a membrane around it. It's terribly thin, a 5/millionth of an inch, but it covers every square inch of every cell. And this membrane has the property of being relatively impermeable to ions. So ordinary ions, which are things like sodium and potassium from your diet, from daily salt, those things ordinarily cannot get across a membrane. It's an impermeable barrier. But there are ways in which cells have contrived to move ions across. Cells can make pumps that are just what the name suggests. They actually do work pulling ions across to one side. They use the cellular energy reserve, ATP, which is made from the glucose in your diet. So the idea of this is it's like charging up a battery. If you push all of the ions over to one side, say the left-hand side, you've done some work, you've stored up energy, and you can then release that energy for signaling purposes. This is what our brain, this is where our brain consumes most of its energy. A brain runs at about the same power as a light bulb, a 20-watt light bulb. And most of the energy is involved in what I just showed you, pumping ions across the membrane to one side and storing up energy. So the energy is released by the activity of ion channels. An ion channel is just a little pore, a protein molecule with a hole through it that can allow ions to run out. But this pore is closed by a gate, this yellow thing here, so that the channel doesn't have to let ions run all the time. It can be turned on or switched off. When the channel is turned on, which can be done by several different mechanisms, voltage, nerve transmitters, mechanical stimulation, or whatever, now ions can run through, some of the electrical current flows out, and that sends an electrical signal in the cell. So the battery is charged up by the pump, and it's discharged by the flow of ions out. Now, this flow of ions produces something called an action potential, which is the special nerve signal that propagates along the entire length of the cell. And this is a sort of frightening diagram of what the action potential comprises. The idea is that inside the cell normally are potassium ions. They're the blue ones, as mentioned down here. When an action potential comes by, the membrane has ion channels that suddenly let sodium, which are the orange ions, run in. And those positive ions make the interior of the cell more positive. A little later, the potassium ions run out, also through a set of ion channels, and that makes the interior more negative. And then the whole thing is repeated again and again. But the important point is that this sends an electrical message. It's rather like a little blip running down a wire or a bit of cable carrying a computerized message. In this case, the whole action potential slides down the axon to its tip, carrying information from one end to the other. This wastes a certain amount of energy, and the cell has to build up its energy reserves again by using glucose to make more ATP, pumping more ions. And so when we're busy and thinking hard, we burn off energy, then we recoup it, we burn it off, we regain it, and so on again and again. And it's rather like a bank account. You draw down the bank account for a big expenditure, then you build it back up, draw it down, and so forth and so on. Individual cells are doing the same thing. Now, when the signal reaches the end of the axon, it's got to be transmitted to another cell. And that's done at what's called a chemical synapse. So here's the end of the axon, and at its very end, the synaptic terminal, there are little bags of a chemical called a neurotransmitter. There are various kinds. You may have heard of acetylcholine, dopamine, or other things. But these are all chemicals that are released from the little bags when the cell is excited. They then fall through the space to the adjacent cell, the so-called postsynaptic cell, and they then excite it. This is where our various pharmacological agents act. For example, antidepressants or almost anything else you can think of. They tend to act on the functioning of these synapses. The importance of the synapse is that this nerve cell that I've shown here in blue is not receiving just the one input. It's receiving tens of inputs, hundreds of inputs, thousands or hundreds of thousands of inputs. So the computation is done at this level. Each cell is looking all the time at all of these inputs. It's weighing them and then deciding whether it should be excited or not. And if it is excited, then the flow of information here across the synapse is propagated into this cell, which in turn becomes active and passes on the message further down the line. So this is the basis of all of the signaling in our nervous system. And here's a very schematic diagram showing how the information flows. Imagine that light comes in the eye. It would excite this particular cell, which would send a message through this one to this one, finally excite this cell. It sends its axon down the spinal cord. It excites a motor neuron, the dark red one here, which causes a muscle fiber here in the arm to move and maybe a finger to twitch. Or you might hear a sound, and I'll introduce this cell in a minute. It sends information through this channel, again down, and causes, say, a reaction like twitching a muscle. The same thing happens in the heart. The lub-dub stem from the activity of ion channels and the same sort of signaling in the muscle cells that make up the heart. Now, to get an input into the system requires that we convert information from the external world into an electrical signal that the brain can then interpret. And that's the process of sensory transduction. And I'll give you a very brief example of this dealing with the sense of hearing. This represents a bit of speech, 2.5 seconds in length. Up here, you see an electrical signal of the sort you would record if you looked at what's coming out of the microphone that I'm now wearing. And below, you can see the syllables of speech broken down by frequency. So these are low frequencies, these are progressively higher frequencies or higher tones. So this speech has some relatively low frequency parts, it has other chirps that are at higher frequency. And this is actually a bit of a poem. This is The End of Fern Hill by Dylan Thomas. As I was young and easy, in the mercy of his means, time held me green and dying, though I sang in my chains like the sea. This piece is though I sang in my chains like the sea. And you can see that the low frequencies are the vowels, like though and sang and so forth. And the high frequencies are the consonants, sang, chains, sea, and what have you. And the ear has to constantly capture this stream of complicated sounds, break them down in terms of their different frequencies, and then tell the brain what it's hearing in order for you to understand speech. The cell that does that is called the hair cell. It isn't a neuron, but it's a smooth cell that does make synapses at its base. And the thing that gives it its name is this so-called hair bundle, this little cluster of feelers sticking out of the top. And you can see that in a different kind of microscope. This very elaborate and elegant structure that's somewhat like a set of organ pipes. It has a short edge and then grows progressively taller as you go across. It has something like 50 to 100 pipes in the particular organ. This device is mechanically sensitive. So each of the hair cells in your ear responds when a mechanical stimulus reaches it and tweaks this organ pipe array back and forth. So each hair cell is a mechanical receptor. The sound is converted into vibrations. The vibrations then cause the cell to be excited. And the way in which the cell is excited is simplicity itself. At the top of each of these little hairs, there's an ion channel. So 1, 2, 3 of them. They're tied to little strings that are made out of protein. When the hair bundle is pushed, you can see that these little springs will be stretched by the sliding between the successive rods. And that stretch pops the channels open, and just as you saw, ions will now flow in through the channel, changing the voltage, the electrical signal inside the cell, and that information will be propagated. Now, the organ that breaks down sound into its constituent frequencies is the cochlea, the name coming from the Greek for a snail. And the cochlea can be simply thought of as being a long tube, about 1.5 inches long, filled with liquid, and with this elastic structure, like a rubber band, running down its length. When you hear a sound, the sound pushes and pulls on the eardrum, and the 3 little bones of the middle ear, which you remember from somewhere in high school or junior high, those 3 little bones move back and forth, transmitting information into the cochlea and causing this rubber band-like structure to oscillate up and down. The thing that's special about this membrane is it isn't like a guitar string or something that's homogeneous, the same everywhere. It's sort of a magical string. At this end, at the apical or top end, it's broad and floppy, and at this end it's very taut and very narrow. And as a consequence, different frequencies of sound excited in different places. So low frequencies make it vibrate up here, high frequencies make it vibrate down there, and other frequencies are represented in between. And the really important thing is that if you hear a complex sound like my voice, it sets the basilar membrane, this elastic structure, into vibration in multiple different places, corresponding to the different frequencies you're hearing at any instant. So this device is sort of the inverse of a piano. In a piano, you have a series of separate strings, each carrying a particular note, and the sound that comes out is the combination of all of their activities. In this case, we are taking a complex sound like the voice. We break it down into different components, representing each of those in a different place. And hair cells in those different positions then report what is being heard. Now, since we're in the Upper East Side, it seems worth mentioning this other snail-like structure, the Guggenheim Museum. The Guggenheim Museum, in this map, is at the end of 88th Street next to Central Park. And it turns out that one can make a nice simulacrum of the cochlea by imagining that one could unroll that. And if you did and stretched it all the way down 88th Street to Carl Schurz Park, you would have something that, like the cochlea, has different frequencies represented at different places. So the cochlea has high frequencies here, progressively lower ones as you move down. And this very much mimics the price of apartments. There's some extra zeros that you can just barely hear, but just barely see. But if you want to live here, you're going to pay 20 million, 2 million, 200,000 or so. 20,000 sounds very cheap, but that's the squirrels in Carl Schurz Park. So what happens when you hear a complex sound, like the word chains, is that there is a series of different oscillations along this membrane, chains with relatively high frequency, the anes part with lower frequencies, and at the end with high frequencies. And each of those excites particular hair cells lined up along the road, along this membrane, and those in turn signal by these blue nerve fibers, and actually the active nerve fibers are red in this diagram, and that carries information into the brain about what you've heard. Okay, so the next issue, having introduced how the signaling works, is how things get that way. How do you put the brain together? And there are a series of steps that are highly choreographed by genetic information and that are key to brain formation. The first of these is growth, obviously. And the brain starts out just peanut-sized as a little layer of cells with liquid inside. And like this glove, it gets sort of inflated by the liquid, but it has just a very thin wall. The cells in that wall then undergo a series of divisions. So as time goes on, the wall gets thicker and thicker as new cells are added. So as cells are piled on, it gets thicker, like this. This isn't easy with a rubber glove, by the way. And it gets thicker. So layer after layer piles up until you get something that is more or less brain size. Now, the most remarkable thing about this apparatus, after it's assembled, after all the cells are piled in there, is that the cerebral cortex, which, as I said, is the really important part, has to be crammed into this tight space. The cerebral cortex is where we do our thinking, and we need as much of it as possible. But this is a very tiny object that has to be crammed into a head, to a skull of a fixed size. So what nature has done is folded the brain. It's corrugated the brain. So just as a way of reminding you of this, this gives you an idea of how big the cerebral cortex really is. I don't have a pizza, but I have a piece of aluminum that's the size of a pizza. That's how big your cerebral cortex is, right, if you fold the thing up. So this is corrugated in the way that we see. It's folded over the brain like that until you finally get this corrugated rough structure cramming the size of a 16 or 20-inch pizza into a much smaller container. The next issue is pathfinding. The nerve cells that have been created have to somehow get to their appropriate targets. And the way this is done involves a whole set of chemical signals that we understand in part. In fact, Marc Tessier-Levin here is a person who's contributed in an important way to our understanding of how this comes about. So here's a schematic diagram of the brain, similar to what you've seen before. This represents the cerebrum, the cerebellum and brainstem, and here's the spinal cord. So let's take a very simple example, something like a muscle, perhaps the muscle that contracts when a doctor taps on your knees and makes a reflex occur. There are small muscle fibers that are sensory in nature. They are innervated. They receive a nerve input from nerve fibers that wrap around them. Those nerve fibers then go into the spinal cord. Here's the nerve cell and its cell body. Once it gets into the spinal cord, the nerve fiber has to find its way to the appropriate target. It uses a set of chemical cues. It uses gradients of a sort that I'll show you in a few minutes, and various other kinds of information. It knows that it should grow up in this direction, end at this point, and contact another cell. This cell has a different set of motivations, a different set of chemical sensitivities. And it does something interesting. It crosses the midline, and we know something about the factors that cause it to cross the midline. Moreover, once it crosses the midline, it loses all interest in going back. There's a switch that's thrown, a new set of genes are turned on, and the cell now progresses up on this side and will never cross back again. And finally, there's another synaptic connection that sends information all the way up to the cerebral cortex. So this is where, for example, you might actually feel a response as a result of this tap on the knee. At the same time, information needs to flow in the other direction to cause a motion to occur. So we have other connections. We have neurons up here in the brain that can send a message down to cause a contraction. So this message flows like this. It also crosses the midline and then runs with a separate set of chemical cues down this far. It then makes a contact with another neuron still, and that last neuron is the motor neuron, so-called, that actually sends a signal out and makes the muscle contract. There's also a direct connection between the first cell, the blue one, and the motor neuron. So this is another synapse. When you're tapped on the knee, this signal goes right through the spinal cord, comes right back, and causes your knee to twitch. But at the same time, the information goes up to your brain and tells you what's happened. And at the same time, with the green neuron, you can turn the system on and voluntarily move that muscle. It isn't just driven by reflexes, but you can make it do what you want. So this is something that's still being worked out. We want to understand what all these chemical cues are. And of course, as was mentioned in the introduction, those cues are very important when something goes wrong in the nervous system. Various diseases can disrupt the process, and we'd like to understand what goes wrong and what can be done to prevent or reverse those problems. The next thing is mapping. We have a prodigious number of neurons, as I mentioned, billions and billions of them. How do you make all of those connections? Here's just a particular example in the visual system. Visual information flows to the so-called visual cortex at the back of the cerebrum, and there are a number of separate areas there. V1, the first visual area, V2, V3, V4, and so on. And then other areas with names like middle temporal, and there's yet another one here that we're not seeing very well. There's a purple area down here that has another name. Those subserve different functions. So the first visual area, V1, looks at oriented edges. It makes sort of an impressionistic or even cubist drawing of the world around us. It tells us where all the edges of things are. The second area, V2, is very good at parsing stereoscopic depth. The third area, or the fourth area, V4, looks at different colors. This purple area that you can't see so well notes faces. MT detects motion, such as this falling feather. So the information that's coming in is spread sequentially through each of these areas, not directly to the next, but one to another in various combinations. And each of those areas has to conduct a separate set of calculations and has to adduce particular things about the environment around us. The same thing occurs in hearing. Information flows to the first auditory area, then to various areas that are still uncharacterized around it. Some of that information goes to Wernicke's area, and this is the important part of the brain that parses our speech. It breaks down the sounds that I showed you before and decides what syllables are what and what a person is actually saying. And that area is highly connected to a motor area, Broca's area. This is the region in which, at least in part, we generate speech, in which we generate responses to what we've just heard. So how do you make connections among this huge, huge number of cells? Here's a diagram showing what the problem is. There are various levels. This might be the first, the second, third, and fourth visual area. Each of them contains hundreds of millions, if not billions, of cells, and they've all got to be connected up in a highly specific way, layer after layer. Not only is there this hierarchical organization from lower to higher areas, there's also a lot of parallel processing. That is, information branches off from one pathway to others and to yet others. So at this level, there are three different separate things going on. This might be color processing, this might be motion, this might be stereoscopic depth. How do you make all those connections? So here's sort of a diagram of what goes on conceptually. This represents the lower part of Manhattan, this represents Brooklyn, and each of these is a street corner, say First Avenue and First Street. And let's suppose for whatever reason, and I don't know what the motive might be, but let's suppose you want to connect these things together so that you would connect First Street in Manhattan and First Avenue to First Street and First Avenue in Brooklyn. You would put some sort of a wire between the two and go on and connect all the other corresponding points in this array. This is what you would get. It's something that looks just like the nervous system, this complicated set of connections. The problem is, to do it this way, corner by corner, would require a huge amount of information. It would require 1,200 wires, but more importantly, it would require specifying each of 2,400 different corners and connecting them up appropriately. The brain does not have the genetic information to do that. It can't specify each of billions and billions of neurons. It must have more general rules. And in a way, this is a little bit like what goes on when you build a house, right? If you have plans for a house that your architect has drawn, those lay down where the walls should go within an inch or two, and they say where the electrical amenities should be and the like. But they don't specify where every stud is or where every nail is to be driven. And the nervous system has to work the same way. It has some broad blueprint, but then it has to make decisions, cell by cell, about the finer wiring. So how can you solve the problem? And here's a way of thinking about it. Instead of specifying every street corner, the brain specifies just a couple of axes or dimensions. And I've arbitrarily made them brightness. So this end of Manhattan is darker, this end is brighter, and yellowness. So this end is more yellow, this end is less yellow. So this might be because something yellow is coming out of the Hudson, which seems plausible, and sort of oozing across Manhattan. But it's setting up a gradient, right? And similarly, you can do the same thing in Brooklyn. And the idea is then that you connect similar points. It's hard to see, sorry. But the idea is that the brightest, the darkest yellow point is connected to the darkest yellow point. The brightest yellow point is connected to the brightest yellow point. The point in the center is connected to the point in center, and so on. And by doing this, you can make all of these connections just by letting every nerve cell here grow to a corresponding position over here. But now you don't need thousands of pieces of information. Just these two gradients, just these two shadings of brightness and yellowness is all you need. And this is, in fact, the way the nervous system works. So here's a projection from the two eyeballs back to part of the brain. And exactly this sort of arrangement works. There's one chemical that's shaded in this direction. Nerve cells use that information to go to the other side of the brain and to wire up in corresponding positions along the same axis. And the side-to-side positions of the retina have another corresponding gradient of intensity that the nervous system uses. So each cell can find its appropriate position and make the appropriate connection. So with using just a few chemicals, you can specify how billions of cells can wire themselves up. The final important thing that goes on is competition. Nerve cells fight with each other. They fight over their targets. So here's an initial configuration with 3 nerve cells, or would-be nerve cells, trying to innervate these 30 different nerve cells, the blue ones down below. And depending on how things start, when the different cells are born, and so on, some of them do better than others. So this orange cell, for example, is not being very successful. And what typically happens in that case is that the cell retracts its axon and often goes ahead and dies. Similarly, if a cell is not innervated by anybody, it will often die. In fact, in much of our nervous system, there are twice as many cells as we need, and during youth, the excess number are trimmed away by this kind of competitive process and by cell death. So when things work right, you end up with a neat arrangement in which each of these 2 surviving cells encompasses about half of the array of the postsynaptic target cells, and they're more or less equally shared. There can also be problems if one of these cells is at a comparative disadvantage. For example, in this case, we have 2 cells that start out innervating about the equal number of targets. But suppose something bad happens to this cell becomes somehow weakened, then the yellow cell tends to expand farther and farther, the red cell gives up territory, and again, it may ultimately fade from view. And this is what goes on in something like the problem of amblyopia, or what happens with lazy eye. As you know, if a child has an eye that deviates, that eye often is weakened in its connections to the brain. The eye functions perfectly well, the nerve fibers function well, the information flows into the brain, but as with this red cell, the fibers are basically disconnected, and the other eye, here represented by the yellow cell, takes over. There was a former president of this university, Torsten Wiesel, who many of you know, who, with his partner David Hubel, worked out a good fraction of our understanding of this competitive interaction. And what they were able to show is that there's a so-called critical period, a narrow window during the first months or first years of life when this kind of competition goes on. And it's key that during that period, one see to it that the weakened eye be given an advantage. For example, one can do surgery to repair an eye that's deviating, and one can then patch the other eye, making the yellow cell less competitive, allowing the red cell to reassert itself and to produce a normal set of connections. Finally, we come to the issue of genetics. As I said at the outset, all of this is encoded by a small number of genes, just 20,000 or so. And those genes shape us in every way. They have an enormous influence on who we are and what we can do. What does 20,000 genes look like? What is that information? It's about this much. Here's a medium-sized library. If each of these books has about 200 lines per page of small type, like that in a telephone book, this is the size of all your genetic information. So on the one hand, it's imposing. It's a lot of books, a lot of small print. But on the other hand, it's a number of books that we can readily read in a few years' time. And in fact, this has, of course, all now been parsed and is available on disks. Every single letter in every single one of those books is, at least to some precision, now known. Now, it's interesting to ask what happens when a mistake is made, when there's a typo in one of these books. And I will attempt another demonstration to give you an idea about this. This demonstration has to do with color vision, and you'll see what's going on in a moment. I hope I won't be in your way. Shh. I apologize. This machine is getting old. Slide projectors are nearly extinct, as you might suppose. Okay. So what we have here is, first, on your right, a yellowish-orange color. There's a pure yellow-orange being shown on the screen. On the left, there's a combination of two different colors. And that combination of two colors looks like this. If I displace it, you can see there's a pure red and a pure green. And we've learned somewhere in junior high school that red plus green makes yellow. So the object of this exercise is to match the two, to control the amount of red and green until they match. So one can turn the knob here, make it greener, make it redder, and somewhere along in here there will be a match where they look more or less the same. They won't look exactly the same, but as close as they can get. Now, I'm cheating. I look at the knob and I know sort of, according to Kathy, where they should match. If I do this match myself, I get this. I hear snickering in the back. That's about the best I can do. Those two things match for me. I dare say I'm in a minority. Leslie is laughing at me. Would you care to come up and do a match for us? All you have to do is turn the knob. So just turn this one way or the other, and it'll get redder or greener until it looks about the same. What do you think? I mean, is that better than mine, or what? Yes. Wait, don't go away yet. You get this valuable Rockefeller University coffee cup. Give it to your mother. Now, the question is, does anybody else agree with me? I don't know if anybody cares to say, but there may be other people in the audience who disagree with the majority opinion. Anybody willing to admit as much? Maybe not tonight? 5% of men have conditions similar to mine, and they will match with varying degrees the red and green in different ways. Let me show you what this is all about and what it has to do with genetics. I'm a mutant, you see, and here's what's happening. 30 million years ago, our ancestors in Africa had one gene that controlled color vision. And 30 million years ago, the old world apes, including ourselves, had a duplication of the gene. It was copied twice, and then it diversified into making a gene that's sensitive to red and a gene that's sensitive to green. They both sit on the X chromosome. Now, the X chromosome part is important because, of course, women get 2 X chromosomes, men only get 1, and that makes a difference in what happens as a result of this. So, normally, when eggs and sperm are being made, there's something called crossing over. So here is the red gene and the green gene, say from one's father. Here are the same genes, say from one's mother. And as an egg or sperm is being made, the DNA from this one combines with the DNA from that one, and this crisscrosses the other way. So this becomes the new combination, A, B prime, green, A, B prime, green. And this becomes another combination, B, A prime, green, and so on down here. So this is fine. It just assorted the genetic information, but all the information has been preserved more or less in normal form. But mistakes can be made. These two genes, because they diversified so recently from each other, look a lot alike. And sometimes the copying process gets confused. So it makes a crossover like this one, in which you have part of a red gene hooked to the green gene instead, and part of this green gene hooked to the red. So let's see what the product is. This gene now becomes half red, half green, and that's all. This gene has red, half a green, half a red, and green. It looks like that. An individual like this, which is about 5% of the population of men, is red-green colorblind. He cannot tell the difference between red and green, even though, and you know, looking at this picture, it would be very hard to tell that there are two parts to that arrow. The perhaps luckier individual, like myself, gets this combination. I have a normal red gene. I have a hybrid gene that is half green and half red, and I finally then have an intact green gene. I should turn this noise off. So as a consequence of this, the protein made from this gene does not have enough green sensitivity. I have to use a lot more green light to make a match. So my match looks to you perhaps lawn green. Her match looks to me more or less cherry red. There's that much difference in the two. So the point of all this is not just to make fun of me or of you or of anywhere else, but the point is that this shows how evolution actually goes about its business. This kind of mistake occurs not infrequently. In fact, going back to this point, genetic duplications can often occur. Usually when these genetic duplications occur, they're inconsequential. The second copy may be deleterious, it will then be weeded out, or it may be neutral, it may have no impact at all, in which case it'll hang around, but it'll just be converted into junk in the genome. Next, this type of crossing over is a normal mechanism, and this crossing over allows the assortment of all kinds of different genetic combinations. It means that every child who is created has an absolutely unique combination of genes contributed by one parent, by the other parent, in all kinds of complicated mixtures. This means that each child is sort of a separate story. Each child may have extraordinary abilities that will then, we hope, get fixed and be propagated through the rest of the population. And finally, mistakes get made. So here's a mistake, a mistaken crossover. Genes get lost, genes get duplicated, genes get mutated in other ways, and this constantly is affecting all of us. Most of the results are inconsequential. So all of us have a number of genetic mutations. Some of them are very small changes that allow us to do things such as DNA sequencing to recognize criminals and the like, but they have no effect at all on one's life. A few of these mutations are beneficial. And again, one hopes that they get fixed in the population, that they get preserved and propagate. And a few of them are problematical. They lead to sickness, they lead to death. And this is something inexorable about this process of reassortment. So I guess the final point I want to make is that in this country we have enormous advantages. We have terrific equality. We have an enormous and unparalleled range of opportunities before us. But we also do face some genetic determinism. Each of us is dealt a particular hand genetically. Some people are born with exceptional abilities. Some people are born with really crushing disabilities. And if indeed a society is defined, if it's evaluated by how it treats its most vulnerable people, it's important to remember that. So I thank you for your attention.
Thank you, Jim, for a fascinating lecture. I'd like to open the floor to questions, and there are 3 roving microphones. I'd ask that you wait for the microphone so that everyone can hear the question.
Thank you for an extraordinary lecture. I noticed that the powerhouse in the brain seems to be glucose, and maybe there are some suggestions for people's diets that might increase our brain power.
Unfortunately, I don't know of any. Glucose is a major source of energy. The brain can also burn fat, which doesn't help dietarily. But obviously, keeping the brain going is a primary responsibility of the body. So basically, the brain has unusual metabolic capabilities that keep it fired up even after everything else is running out of energy, because it's absolutely necessary.
Why the crossover?
So the crossover assorts the genetic information and mixes it. This is what sex is for. One thinks of it as just a chore, but
Different crossover. Why the crossover from...
this crossover.
Yes.
Okay. Let me finish answering the other question while I'm at it. Since I've gotten that far, the crossing over in genetics is what stirs things up together and allows genes that were separate in the parents or in the grandparents to be recombined in original and new ways. And so that's what really stirs the pot and produces all the new genetic information. Why the crossing over, nobody knows. This has been a problem ever since neuroanatomists identified it. It makes no particular sense. There have been many hypotheses. I don't know if Mark has any of his own, but there's nothing intrinsic about the nervous system that says that information from your right side should go to the left side of your brain, and correspondingly, that action originating in the left side of your brain should end up moving your right side. So it's imponderable. Sorry.
I was really curious about what you said about the mutations that have some benefit and some have no benefit and some just kind of sit there. Could you comment on some of the mutations that we're aware of in populations, for example, the asparagus issue, that you can smell asparagus, the issue of some people can taste cilantro in a way that tastes like soap, and it seems to be consistent within the population that hates cilantro. Can you comment on those? Are those simply useless evolutionary advantages or disadvantages or small mutations?
Yeah, no, so you're exactly right. Those things do represent what are called different alleles, that is, different flavors of the same gene. And our olfactory system, our smell apparatus, is a particularly striking example of that. We have something on the order of, I believe, 600 active, 400, I'm told, active odorant genes. Mice and rats and so on have 1,000. They are really very olfactory animals. Their life depends upon their olfactory capabilities. We've lost more than half of our genetic complement. We have a lot of what are called pseudogenes, genes that have been corrupted because they're no longer necessary. So in addition to those corrupted genes, we have, as you said, different alleles, different flavors of genes that make us sensitive or insensitive to various things in the environment. And those taste examples and the smell examples you mentioned reflect exactly those differences in two proteins. Now, we have in each of our genomes a number of genes present in 2 copies, one of which is mutated in such a way that we would be dead if we had 2. So genes such as that for cystic fibrosis have a prevalence of about 5% in our population, quite a high percentage. It's only if one has the bad luck of getting 2 copies that the disease is apparent, but the disease is there latent in anybody who is heterozygotic. In many cases, having a heterozygous state for a gene that may be deleterious may confer some advantage. It's thought, for example, that the cystic fibrosis gene in people who have 1 normal copy and 1 abnormal copy may be somewhat more immune to various kinds of enteric problems that were an issue for Northern Europeans. Similarly, for African Americans, as you know, there's sickle cell disease. Having 2 sickle cell mutations can be deadly, but having only 1 confers immunity to malaria. There's similarly in the Southeast Asian population, was it glucose 6-dehydrogenase, when mutated in 1 copy, it protects against malaria, but 2 copies can be deadly. So we are all carrying around this combination of mutated genes, of genes that are in the process of becoming different things. And again, this is assorted in each generation. Some people have good luck and some people have bad luck in terms of what hand they're dealt.
Hi, can you share a little bit about some of the research you're doing and some of the, especially for the regrowing of, let's say, the hair cells for neurosensory or hearing loss?
Sure. The 2 things we're interested in are, first of all, how does this thing work? It turns out that our ears are not just passive systems for catching sound. The ear actually has an amplifier, a biological hearing aid built into it. And our hearing is about 100 times more sensitive than one would suppose just from the physics of the situation. It turns out that this hair bundle doesn't just respond to oscillation, to, say, sound, but it can actually vibrate on its own, and human ears, normal human ears, can produce sounds that come out of the ear. 70% of normal ears do that in a quiet environment, which seems quite weird. But really what's happening is there's an amplifier in there, and that amplifier is just like the public address system in this room. If we turn it up too far, it begins to oscillate or howl, and the sound comes out. So we're trying to understand how that works. The other thing we're trying to understand is why the ear is so vulnerable, and what we can do when these cells are damaged. How can we prevent that, or how can we replace them? Normally, these hair cells are not replaced. You're born with 16,000 in each ear, and loud sounds, certain chemicals, aging, all whittle away at them, and we progressively lose our hearing. We would like to find ways of bringing those cells back, perhaps by using stem cells, which we're studying in zebrafish of all things, trying to learn whether it's possible to regenerate the cells and to regenerate similar cells in our own ears.
I wanted to ask a question about the competition among the cells and the connections that are formed. You mentioned that that competition goes on for a period of time, but I wasn't clear whether that was months or years that the cells were competing with each other to make these connections. And as a kind of a follow-up to that, what happens when, is there a way to reverse when one aspect of the brain is becoming more dominant over the other aspects? Is there a way to kind of retrain it to?
Yeah, they're really good questions. And the answer is there are different critical periods for different faculties. So that for vision that I mentioned, left versus right eye, extends over a few years of time. So children up to an age of 3 or 4 can still have some or complete remediation if they get the appropriate eye operation and have an eye patched. There are other things that we know occur earlier. So for example, if you look at young children, every child in every culture, everywhere in the world, can understand all speech sounds. So Japanese children can understand the R-L distinction. Our children can understand Mandarin Chinese tonality. People can understand Xhosa and other languages that have a click, which I can't do, Xhosa. By about 6 months of age, children begin to lose that. And by a year, they've by and large lost it altogether. So one loses the sensitivity to sounds that are not in one's native language after a critical period extending just 6 months or so. One can obviously learn foreign languages later, but it becomes progressively harder thereafter to learn it in an automatic way.
Just on a practical note, what are the chemicals that affect the ear hair?
So there are several things that affect these cells in a reversible way. Large doses of aspirin, which people used to take for arthritis, and quinine, which they used to take for malaria, both cause this thing to oscillate spontaneously and give people tinnitus or ringing in the ears. But the things that devastate it are two classes. The first are antibiotics called aminoglycosides. This is gentamicin, tobramycin, neomycin, drugs of that sort. Those devastate the internal ear, but they're sometimes necessary to treat bacterial infections. For example, endocarditis, a life-threatening infection of the heart. You simply have to, as a clinician, use those drugs to save a life, but you may cause partial or complete deafness as a result. The other major one is cisplatin, which is the major chemotherapeutic for ovarian cancer. And again, there are many women who have lost some or all of their hearing, but had their lives spared by the use of that drug.
On a evolutionary timeline, what do you say is next?
I think probably the next stage is going to be a reception in the lobby. What do you think?
I think that's a great idea. But maybe we can take just a few more questions. We have one there. Yes.
What about the effect of hormones on hearing, testosterone, for example?
Yeah, so there are some interesting effects, even effects on the sounds that come out of the ears that I mentioned. At different stages during the menstrual cycle, women have different degrees of perfect pitch. Women musicians notice this. Also, homosexuals have somewhat different sounds coming out of their ears. I mean, one does not know why, but some difference in brain structure causes these sounds to be modulated differently in men, women, and in homosexuals of either sex. So we don't know the basis of that, but there are definitely hormonal effects.
Again, on the competition of the brain and what it goes inside, it seems that there are different phases for this. Is there any applications in education for this? Like, for example, there is a phase where children are better suited to learn something as opposed to another phase.
Yeah, I don't think we know enough about that. Maybe I'll ask the expert. My wife is a developmental pediatrician, and she's shaking her head. So I think the answer is we simply don't know enough. I would say there almost certainly must be, because these things do have critical periods. There are particular things going on at particular times. And it's striking when you're raising a child how from day to day things will switch. You can almost hear the genes clicking as a child's behavior changes radically for better or worse. And new habits are adopted, old habits simply evaporate. So during those critical periods where the changes are occurring, it's no doubt there could be ways of accentuating the good, maybe suppressing the bad, but we just don't know enough yet.
I wonder, there are a lot of children who suffer from sensory integration disorders, and I wonder, if you're a researcher, you have theories on where that issue is coming from. Is it pathfinding, mapping, the competition, and if you have theories or thoughts, thanks.
Maureen. So this is Maureen Packard, my wife, who is a developmental pediatrician, and she is here to answer the hard questions.
We really don't know. Clearly there are children who have sensory issues, which means that they process sensations coming in differently than the rest of us. So for some of these kids, sounds can be really feel horrible. For some of them, certain textures on their skin is really aversive, but we don't know. There is a degree that you can sort of retrain, and certainly the occupational therapists who do things like the brushing and rocking, and it makes it better. But we don't understand why those kids have those issues. And it's something that is not just brand new, because my husband had issues like that when he was a kid. I know I did. I think all of us have them to some extent, but for some kids it becomes more impairing.
You can stay here in case there's another hard one. Let me answer one question that came from a gentleman at the back, and I didn't give an answer to the second half of it, but I think it's an important one. And that is there any way of replasticizing the brain? If there are critical periods that are passed, if the eyes have not been properly wired, can you have a second chance? And the answer so far is no, but it's clearly an area that neuroscientists are interested in exploring. We would very much like to find a pharmacological means, I'm sure your erstwhile company has looked into things like this, of adjusting the brain and allowing connections to reform. Lower animals, so-called, can do this very well. Even if nerve cells are destroyed, they can sometimes replace them, or they can at least regrow the connections. We can do this to a limited extent, and this is what is involved in, say, the recovery after a stroke. Some part of the brain may be damaged, but other nerve cells nearby will grow into that area or nearby areas and establish a new set of connections. But obviously our plasticity is limited. There's a point beyond which we can't recover, and that's what we would like to correct in the future.
Perhaps you could comment a little bit on the process of neural recruitment. My understanding is that it's reported that when people early in life lose sight, that sometimes their hearing is enhanced. At least that's a report. I don't know if that's actually true, but many, interestingly, piano tuners have been blind. And I wonder if you could comment on that process of the neural recruitment.
Yes, so that is somewhat related to the foregoing question. As I mentioned, when a stroke or the like occurs, there can be rewiring. Something similar happens if early in life some particular portion of the ordinary connections are not present. So, for example, in a blind person, the visual cortex may be perfectly intact, even though the eyes are somehow non-functional. And it's been shown using fMRI, functional magnetic resonance imaging, that other functions can be taken up by the visual cortex. It's also been shown in animal studies that if you take the information that normally is connected to the, say, visual system and connect it to the auditory cortex, you nonetheless get some proper function, or vice versa. If you send auditory information to the visual system, the brain still knows something about how to process that. So the recruitment no doubt does involve the extravagant growth of nerve fibers to places that are not getting a proper input, and there is some expansion of the faculties as a consequence of that.
Could you comment on what you know of the utility of EMDR used in treating trauma, eye movement desensitization and reprocessing?
No? I'm
No.
afraid not. No, I actually don't know the treatment. I apologize.
Okay, for the next lecture then.
Okay, done.
Just wanted to ask in regards to nutrition, vitamins, this and that, in regards to the brain. Is there any correlation to help, to enhance, to make changes?
So this is certainly an area that a lot of people have worked on, and it's an area where Maureen has a strong opinion. If you had breakfast with us, you would find a little heap of pills next to the cereal. So you can tell us what we know about that.
We actually don't know as much as we would like to know. Clearly, humans have been around for a long time, and given our diet, it clearly is enough to make your brain grow and develop. We do know that if children are iron deficient, that decreases their IQ later in life.
Folate.
Folate can cause problems. There's a lot of interest these days in omega-3 oils, which clearly have in animal studies have effects on the membranes, the coatings of the nerve cells, and help neurotransmission. How those actually help, I mean, if you're already in good shape, does taking more of it make it better? We don't know. I mean, there's a lot of work on people with dementia. You know, is there something you can take that will keep you from your neurons from going bad? But there's no clear answer yet.
Well, I'd like to thank Jim again for a wonderful presentation.